the screen.
Can everyone see it?
Yeah, I can see it.
Yep.
Yeah, I can.
Okay, sounds good.
Okay, hi everyone. My name is Aksati and I'm a sophomore at Notre Dame San Jose High School.
I'm Shika and I'm a junior at Doherty Valley.
I'm Ashley and I'm a sophomore at Mission San Jose High School.
And today we'll be presenting to you our work on using dimensionality reduction and molecular docking to find novel DNA methyltransferase 1 inhibitors for colon cancer. So our overview, first we'll go over a background on each of the concepts and then a detailed methodology, followed by our next steps and acknowledgements. At the moment, we do not have any results. So I just want to make that clear. Okay, so our background. What exactly is DNA methyltransferase? DNA methyltransferase is also known as DNMT, and it's a family of enzymes that catalyzes the addition of methyl groups to DNA. And as you can see to the right, there's a picture of cytosine and methylated cytosine, which has that added group with a hydrogens and a carbon. This process of adding methyl groups is also known as methylation, and it plays a role in gene expression as well as the inhibition of certain genes. However, when DNMT is hypermethylated, or there are too many methyl groups added to the CpG regions, also known as the cytosine guanine regions, it becomes harmful. This can result in the tumor suppressor genes being silenced, and this is a factor that can lead to cancer. I also want to note that there are many DNMTs in the human genome, such as DNMT1 and DNMT3, but we're only focusing on DNMT1 at the moment because we already know that RG108 is an effective colon cancer cell growth reducer. And so we're kind of using that as a baseline to find similar drugs. I also want to note that RG108 is not an FDA approved drug for colon cancer yet, but we already know that it's effective. Okay, so next, molecular docking. Molecular docking is essentially a computational method of predicting the preferred orientation of a ligand to the receptor. There are many softwares out there, such as like RxDock, but we're going to use AutoDoc-Vina. There are other softwares also that are developed to help with molecular docking, such as MolTrans and TorchDrug AI. So on the screen are two images. The first one is DNMT1 when it's visualized in Pymol Viewer, and the CYS1226 residue is selected, which is where we did our, when we were doing our confirmation dockings, this is where we binded all of the drugs to. And the second image is an image of DNMT1 and the binding pockets when we use Pymol.
So to follow up from what Akshithi was talking about, it is important to note that since AutoDoc-Vina only allows PDB-QT files, we need to use certain molecular visualization programs, such as MGL tools, to prepare the ligand and receptor and convert them into specifically the PDB-QT format before we can finally dock the ligand and receptor in AutoDoc-Vina. So the ultimate product that we are looking at from the molecular docking is a list of possible binding allocations of our poses of the ligand and receptor with the binding affinities given to us in kcal per mole. Since our goal is to find the best inhibitor for colon cancer, we look at the best bond, the most stable binding site, and this would be given to us with the binding location with a relatively more negative binding affinity value.
Throughout the process, we also hope to use various dimensionality reduction techniques such as UMAP, PCA, and TSNE. UMAP is uniform manifold approximation and projection, and UMAP defines nearest neighbors as the point which will affect and influence the global, like the influence given to a global versus local information, and minimum distance will affect how compactly packed the local parts of the UMAP plot are. PCA transforms a large set of variables into a smaller one that contains most of the information in the smaller set. As you can see in the diagram, in PCA, the first variable has the most amount of information and it's able to explain for the most amount of variation in the data, and the subsequent components have less information. t-SNE is normally used in 2D, but can be used for any number of dimensions, and t-SNE randomly scatters all points within the chemical space to start a simulation. And the aim of the simulation is to make the point distances match the distance matrix and shuffle points based on how well they match. And t-SNE stops after a fixed number of iterations, but it also stops once the distances between the points have converged. And in t-SNE, close proximity can tell us that a compound is highly related to RG108 and likely to be a good effective inhibitor that can be used in clinical use. So there's a lot of advantages and disadvantages to the techniques that were considered for dimensionality reduction. PCA is good at extracting signal from noise, and it extracts informative dimensions, but the consequences of PCA is that it requires more than 2 dimensions and it's thrown off by quantized data. PCA also expects a linear relationship, which is not expected with t-SNE, and t-SNE also has an advantage that it can reduce to 2D well, like I talked about earlier. But the disadvantages of t-SNE is that it can't cope with noisy data and it loses the ability to cluster sometimes. So the solution to these problems was UMAP, because UMAP is relatively quick. It's a lot faster than t-SNE, and it can preserve more global structure than t-SNE, which you'll see at the end. And it can also run on raw data without PCA processing, and new data can be added to the projection.
Now here's some background on our data set. So before we could cluster the compounds with the use of dimensionality reduction programs, we needed to first have a data set of compounds to cluster. We used over 2 million drugs from Kemble for our data set, and this is pretty much their entire library. PubChem also has a bunch of good compounds in their database, but we decided to just work with the drugs from Kemble for now. After we downloaded the compounds in CSV format, we get a long list of compounds and their properties, which starts from the Kemble ID, which is just the ID Kemble assigned to the compound, all the way to the SMILES, which shows the chemical structure of the compound. We then used code to produce the ECFP of the compounds. And what this is it is the extended connectivity fingerprint, which is primarily just a pattern of 1s and 0s that map the compound's chemical structure, and it is unique to that compound. ECFP is standard and one of the best ways to represent molecular structures in a numerical format when working with a chemical data set. There are many different types of ECFP, and we use specifically the 2048-bit. It is also important to note that there were certain compounds that we had to take out from our data set, but we'll give more details about this in our collecting drugs methodology.
Okay, so now our methodology.
So first we examined various drug databases such as Kemble and PubChem, and we ended up using Kemble because we were able to download all of the drugs in a CSV file a lot more efficiently than PubChem. And then after that, we hope to use some dimensionality reduction techniques such as UMAP in order to find the most drugs that are most similar to RG108. And then once we find those drugs, we hope to dock them using Autodoc Vina and Orca and Avogadro. And from that, we hope to run molecular simulations using tools such as GROMACS to filter for false positive binders. And molecular simulations basically vibrate the individual atom of the protein and the individual atoms of the drugs and see how well they bind. And there are more specific versions of molecular docking. Then we hope to use drug cell line prediction tools such as DeepCDR, which is a hybrid graph convolutional neural network that basically just predicts how well these drugs would work in a cell line using multi-omics data. And then because a lot of drugs are toxic to the liver, then we want to test for drug-induced liver injury to make sure that the compounds that we found are safe for clinical use.
So here is our collecting drugs methodology. In order for us to collect the drugs that we would use to dimensionality reduce, first, as mentioned earlier, we need to create our data set. Our method to do this was to go on drug databases such as Kemble and PubChem to download drug information and download them specifically in the CSV format. We downloaded over 2 million drugs from Kemble and used that as our data set. Our next step was to generate a table for the compounds. Once the CSV file was downloaded from Kemble, we get a long list of chemicals and their properties separated by just semicolons. and quotation marks around each individual chemical property. In order to beautify this, we used code and Excel to separate everything into individual blocks by the semicolon and created a table which is organized in rows and columns. Also taking out the quotation marks for specifically the SMILES column in the process, with the most notable columns for our purposes being the chemical ID in column number 1 and the SMILES in column number 31. After this, we removed the bad lines. And what I mean when I say bad lines, they are the lines of compounds plus their chemical properties, which would give us errors when we try to get our chemical fingerprints. This occurs when only 3 to 4 columns out of the 31 are filled. The rest of the columns are completely empty. And then there were 6,245 of these kinds of lines. In addition to these, there were a few other lines that we had to take out. Some compounds had things like a semicolon inside a pair of quotes. When this is separated for the table, this will result in an error when we try to convert SMILES into chemical fingerprints, the ECFPs. And there were a total of 21 lines that were like this. In addition, there were some lines where the SMILES only had empty quotes. If this were to happen, then the fingerprints we need to produce later on would just be a useless row of zeros. This occurred in 19,991 lines. After we try to use code to remove all of these lines, we would then use additional code to produce a ECFP column in our table from the SMILES using RDKit. And RDKit is just a library of code written in Python that allows people to work with scientific data. The ECFP belongs to the 32nd column, the last column of our table. This fingerprint would then be used, among other things, to be clustered based on known inhibitors. In our particular case, RG108. Actually, could you move back like a slide? Another slide, one more. Okay, so these two pictures here are perfect for comparison. One of them is the CSV file before we actually generate a table. As you can see, everything is in one column, separated by just semicolons. And then the bottom one is after, when the semicolons are removed and each property is separated into columns. As you can see, it is much neater and much more pleasing to the eye than before. Next slide. And then the picture here is a perfect image to show what bad lines are. I know the numbers are really small, but bear with me. And as you can see, the third row and the image there, the properties of the chemical is only filled until the 4th column. The rest is completely empty. The second case is on the row 6 from the bottom. This is what the row looks like when there is a semicolon stuck within a pair of quotes. But the semicolon should not appear within the quotes. So those 21 lines that had this case had to be removed. And then this picture here just shows what the 31st and 32nd columns looks like. And this is the 31st column is the SMILES column, and the 32nd is the ECFP column.
So after we make the graph with UMAP, we hope to see a cluster with RG108 and the similar drugs. We can download the SDF format files from PubChem of these similar drugs and run a series of processes such as geometry optimization, so we can get into the PDB-QT format to run it with Autodoc Vina. And then using the high performance computing cluster, we want to batch dock that with DNMT1 so that we know that which drugs are actually effective with DNMT1. And Autodoc Vina, as Ashley had mentioned earlier, will give us binding affinities that will tell us how well they actually bind. So here is a table of when we docked Diadzine to DNMT1. And this was done as a confirmation to make sure that we were actually using the tool properly. And as you can see, the binding affinities are relatively more negative, like -6 and -5, meaning that Diadzine will actually bind well to DNMT1.
So for our next steps, we first hope to create a script that will automate the download for the file format and to convert the file format conversions for batch docking. And since our current data set only uses chemical compounds, we also hope to somehow figure out a way to use PubChem compounds to expand the number of chemicals in our chemical space. And we hope to use new dimensionality reduction techniques such as PathMap and find a consensus with the drugs that are considered similar from PathMap and the ones that are considered similar from UMAP, so that we know that the ones that are considered similar to RG108 from both of them are likely to work in the clinic. And then we also want to check for drug-induced liver injury. Aside from that, we also want to investigate sharks and whales because sharks and whales are less prone to cancer. So by looking at the protein in their cells, we can find overlapping candidates that decrease the chance of cancer. And so this is like the graph that shows you PACMap and its comparison with the other dimensionality reduction techniques such as TSNE and UMAP. And so this was the original data, and you can see that like UMAP is like the best one for preserving the global structure from these three, but overall PACMap outperforms in performing the global and local structure because it resembles the original data the best.
And finally, we'd like to thank ASDRP for giving us this opportunity, as well as Mr. Kunha for helping us a lot during this project. We'd also like to thank the new group because we had used a Autodoc Vina tutorial from them, and it was really helpful in letting us learn how to use the tool. Thank you for listening, and okay, here are references. And thank you for listening, and we're happy to take any questions now.
Nice work. Nice work. Clinton, are you willing to share that high dimensionality data?
Yeah.
It is ideal for persistent homology and using TDA on that. So I can sense that's going to be a cool project if we use persistent homology. I'm actually for a long time, I'm looking to start a new project using topological data analysis to classify chemical compounds. I have done it on the neurons and neural networks on MRI data. But I think I'm just want to implement it on either chemical compounds, or I thought of speaking to Nelima for collaboration. And now I'm looking at your data, looks like you already have a food ready in your plate.
we're working towards it. You know, we have the general like pipeline. It just we're just kind of, yeah, we're walking towards it, you know. Yeah.
Yeah.
But yeah, for sure. Yeah.
Yeah, I mean, it looks like your plate is ready for homology modeling, and it's gonna be super duper cool. I mean, we can stick to UMAP or TSNA, but TSNA needs refining for that dimensionality reduction. But I think TDA will take a charge of it and take care of it. But we need a higher computational platform to do that.
Yeah, no, I wish, like we swap between Google Colab and the high performance computing cluster depending on needs.
Yeah, I mean, we might, you know, take a advantage of our previous guest speaker, take it to them and ask him as Donald to do it for us, or we can use AWS to run that. But if you are open for collaboration, I have great idea for your data.
Yeah, I know. Yeah, no, I am 100%. Yeah, because I like. You know, our group referenced, we thanked Edward for his expertise for Autodoc Vina. So like what's going on here is very interdisciplinary. So, you know, I'm very open to that.
Yeah, let's use some mathematical modeling and data science, and that's going to be cool.
Does anyone have any questions? We're happy to take questions now.
Good job. Looks like no one has a question. So it's exciting as you're moving forward with this project. Well done.